Hello everyone and welcome to Roadmap. So today's topic is immunoglobulins part 1, and if you like the video, don't forget to click on thumbs up button below. You can also subscribe for more video updates. Topics to be covered are immunoglobulins, that is introduction, then the structure of immunoglobulins, classes of immunoglobulins, then the synthesis of immunoglobulin, major histocompatibility complex, then the immune response, cytokines, therapeutic uses of cytokines, immunity in health and diseases. Now coming to the introduction, what exactly are immunoglobulins? The humoral immunity, which is mediated by a special group of proteins, are called the immunoglobulins or antibodies, and they are produced by B lymphocytes. Now what is the structure of immunoglobulin? This is how a structure of immunoglobulin looks like. Now the immunoglobulin molecules consist of two heavy chains and two light chains. Heavy chain has got molecular weight of 53,000 to 75,000 each, and two identical light chain has got molecular weight of 23,000 each, and it is held by a disulfide bridges, and also the non-covalent interactions. So you can see from the figure that how the immunoglobulin looks like. Immunoglobulin is basically Y-shaped and it is a tetramer. Each heavy chain contains 450 amino acid and light chain consists 212 amino acids. So the heavy chain of immunoglobulin are linked to carbohydrates. So they are called as glycoproteins. Now coming to the constant and variable regions. Now each chain of immunoglobulin has two regions or domains, the constant and the variable. The amino terminal half of the light chain is the variable region, while the carboxy terminal half is the constant region. Now in regards of heavy chain, approximately 1/4 of the amino terminal region is variable, while the remaining 3/4 is constant. Now from like the CH1, CH2 and CH3 are the constant ones. The amino acid sequence of variable regions of light and heavy chain is responsible for the specific binding of immunoglobulin, that is antibody with the antigen. Now coming to the classes of immunoglobulins. Now immunoglobulins have got 5 classes, that is G, A, D and E, which contains the heavy chain. The type of heavy chain determines the class and function of a given immunoglobulin. So the two types of light chain, that is the kappa and lambda, are found in immunoglobulins. Now they differ in their structure in the constant light regions. An immunoglobulin of any class contains 2 kappa or 2 lambda chains, and there is never a mixture of kappa and lambda light chains. So the occurrence of kappa chain is more common in human immunoglobulins than the lambda chain. Now these are the classes of immunoglobulins, that is IgG, E, D, M and A. Now coming to the immunoglobulin G, it is the most abundant, that is 75 to 80% class of immunoglobulin. Immunoglobulin is composed of a single Y-shaped unit, that is monomer, and it can traverse blood vessels readily. Immunoglobulin G is the only immunoglobulin that can cross the placenta and transfer the mother's immunity to developing fetus. So immunoglobulin G triggers foreign cell destruction, which is mediated by complement system. Coming to the immunoglobulin A, it is a single monomer or a dimer or double unit, which is held together by J chain. And it is found in the body secretions such as saliva, tears, sweat, milk and walls of intestine. Now the immunoglobulin A is most predominant antibody in the colostrum and the initial secretion from mother's breast after a baby is born. The immunoglobulin A molecules bind with bacterial antigens present on the body, that is the outer epithelial surfaces, and remove them. And immunoglobulin A prevents the foreign substances from entering the body cell. Coming to the immunoglobulin M, it is the largest immunoglobulin which is composed of 5 Y-shaped units and it is held together by a J polypeptide chain. So it is similar to that of immunoglobulin G. It has got Y-shaped unit and it is a pentamer because it has got 5 Y-shaped units. Due to its large size, immunoglobulin cannot traverse blood cells and it is restricted to the bloodstream. Immunoglobulin M is the first antibody which is produced in response to an antigen and it is most effective against invading microorganisms. Now coming to the immunoglobulin D, it is composed of a single Y-shaped unit and it is present in a low concentration in circulation. Immunoglobulin D molecules are present on the surface of B cell. Their function is not known for certain, and some workers believe that immunoglobulin D may function as B cell receptors. Now coming to the immunoglobulin E, it is a single Y-shaped monomer and it is normally present in a minute concentration in blood. Its levels are elevated in individuals with allergies as it is associated with body's allergic reactions, that is allergies and allergic responses, and the immunoglobulin molecules tightly bind with mast cells which release histamine and cause allergy. So these are the classes of immunoglobulins. Coming to the synthesis of immunoglobulins. Now there are millions of different antigens. It was a big puzzle for a long time how an individual can produce so many antibodies to protect against antigens. So it is now recognized that a gene rearrangement involving a combination of several genes is responsible for generating an extremely large number of antibodies. And this is how the synthesis of immunoglobulin takes place. Now coming to the major histocompatibility complex. Now what is major histocompatibility complex? The major histocompatibility complex, also called as MHC, it represents a special group of proteins present on the cell surface of T lymphocytes. So the major histocompatibility complex is involved in the recognition of antigens on T cells. Now it may be noted that the B cell receptors, which are antibodies, can recognize antigens and on their own, while T cells can do so through the mediation of MHC molecules. In humans, the MHC proteins are encoded by a cluster of genes located on chromosome 6. It is on chromosome 17 for mice, and the major histocompatibility complex in human is referred to as human leukocyte antigen, that is HLA. Now, 3 classes of MHC molecules, that is chemically glycoproteins, are known in human. Class 1 molecule are found on almost all the nucleated cells of the body. Class 2 molecules are associated only with the leukocytes involved in cell-mediated immune response. And class 3 molecules are secreted proteins possessing immune functions. Example, complement components, that is C2 and C4, tumor necrosis factor. So that is T necrosis factor. So this is about the major histocompatibility complex. Coming to the immune response. The immune response refers to a series of reactions which are carried by the immune system in the body against the foreign invader. When an infection takes place or when an antigen enters the body, it is trapped by the macrophages in lymphoid organs. The phagocytic cells, which are guarding the body by constant patrolling, engulf and digest the foreign substance. Partially digested antigen, that is the processed antigen with antigenic epitopes, attaches to lymphocytes. Now T helper cells, which play a key role in the immune response, is brought out through the participation of antigen presenting cell, that is APC. So you can see from the figure that APC is bringing the T cell receptor. That is the T helper cell. So it is usually a macrophage. The receptors of helper T cell bind to class 2 MHC antigen complex displayed on the surface of antigen presenting cells. So antigen presenting cells secrete interleukin 1 which activates the T helper cell. This activated T helper cell actively grows and divides to produce clones of T helper cells. All the T helper cells possess receptors that are specific for the major histocompatibility complex antigen complex, and this facilitates triggering of immune response in an exponential manner. The helper T cells secrete interleukin-2, which promotes the proliferation of cytotoxic T cell to attack the infected cells through cell-mediated immunity. And then the interleukin-2 activates B cells to produce immunoglobulins which perform humoral immunity. So this is about the immune response. Now, there must be a question arising to you that what are cytokines? Now, cytokines are a group of proteins that bring about communication between different cell types involved in immunity. They are lower weight, molecular weight glycoproteins, and they are produced by lymphoid and non-lymphoid cells during the course of immune response. Now, cytokines may be regarded as soluble messenger molecules of immune system. They can also act as short messengers between cells or long-range messenger by circulating in the blood and affecting cells at far-off sites. The function is comparable to that of hormones. Interleukin is frequently used term which to represent the cytokines, and there are more than a dozen interleukins produced by different cells with wide range of function. The main function, directly or indirectly, of cytokine is to amplify immune response and also inflammatory response. So there are some of the therapeutic uses of cytokines. It is now possible to produce cytokines in vitro. So some of the cytokines have potential applications in practice of medicines. Now interleukin-2 is used in cancer immunotherapy and in the treatment of immunodeficiency diseases. Interleukin-2 induces the proliferation and differentiation of T and B cells, besides increasing the cytotoxic capacity of natural killer cells. A group of cytokines, namely interferons, can combat viral infection by inhibiting their replication. So this is about the cytokines. Coming to the immunity in health disease. Now, the prime function of immune system is to protect the host against the invading pathogens, and the body tries its best to overcome various strategies of infectious agents like bacteria or viruses and provides immunity. So, important immunological aspects in human health and diseases, let's learn about it. So, what are autoimmune diseases? The immune system is self-tolerant. Means it is not responsive to cells or proteins of self. So, for various reasons, the immune system fails to discriminate between self and non-self, and because of which the cells or tissues of the body are attacked. So, this phenomenon is called as autoimmunity, and the diseases are called as autoimmune diseases. So, the antibodies which are produced to self molecules are called as autoantibodies. Now, there are some of the examples of autoimmune diseases like the insulin-dependent diabetes, that is the pancreatic beta cell, autoreactive T cells, and antibodies. Rheumatoid arthritis are antibodies against proteins present in joints. Myasthenia gravis, it is the acetylcholine receptor autoantibodies. And autoimmune hemolytic anemia, it is the erythrocyte autoantibodies. Now, what is the mechanism of autoimmunity? It is widely accepted that autoimmunity occurs as a consequence of body's response against bacterial, viral, or any foreign antigen. Now, some of the epitopes of foreign antigens are similar to epitopes which are present on certain host proteins. So it results in cross-reaction of antigen and antibodies that leads to autoimmune disease. Now coming to the cancers. Now growth of tumors is associated with the formation of novel antigens, and these tumor antigens, which are referred to as oncofetal antigens, are recognized as non-self by the immune systems. Tumors have developed several mechanisms to evade immune response. Coming to AIDS. AIDS means acquired immunodeficiency syndrome, which is caused by human immunodeficiency virus, and it is characterized by immunosuppression, secondary neoplasm, and neurological manifestation. Now, AIDS primarily affects the cell-mediated immune system, which protects the body from intracellular parasites like the viruses and bacteria. Now, most of the autoimmunodeficiency symptoms of AIDS are associated with a reduction in a CD4, that is cluster determinant antigen 4 cells. Coming to the organ transplantation. Now, the phenomenon of transfer of cells, tissues or organs from one side to another, that is autograft, or from another organism, that is allograft, is regarded as organ transplantation. So in case of humans, majority of organ transplantations are allografts between two individuals. And the term xenograft is used if tissues or organs are transferred from one species to another species. So, from pig to man, example. Now, organ transplantation is associated with immunological complications and tissue rejection. So this is because the host body responds to the transplanted tissue in a similar way as if it were an invading foreign organism. Now the MHC is primarily involved in allograft rejection, and it is due to the fact that MHC proteins are unique to each individual and the immune system responds promptly to foreign MHCs. So the organ transplantation between closely related family members is preferred, since their MHCs are likely to be closely related and major immunological complications can be averted. So this is about the immunity in health and diseases. Now, I hope that you are enjoying the video, and if you have any doubts, feel free to ask in the comment section below, and don't forget to subscribe and share the video for more updates. Thank you for watching.